Okay, guys, quick question. So did turning the computer around help with the volume? Oh, I just uploaded lecture 7. So please let me know after you listen to it if the volume's any better. I went through it at different points. If you can hear me, I'll try not to go all the way over here. I'll try to stay in this region. Let me know. And worst comes to worst, I'll have to get a mic and be all high tech. Okay. Okay. Just thinking. Okay, so quick business. Think about the oral exam option. Someone already contacted me who has major anxiety issues with tests and thinks it might be a good idea. I think it is a good idea, but you only have one chance to take an oral exam. So think about strategically when you want to use it. Do you want to feel out what my exams are like first and then try the oral exam? Because maybe you're not going to be anxious taking my exam because I'm super laid back and we'll eat candy. I don't know. We'll see. But definitely contact me because I'm scheduling now. So exam 1 is next week. Email me if you're interested. You'll take the exam the same day that you will normally take the exam. So you don't get any extra time. Well, you know what I mean, extra prep time. Okay. Multiple choice questions accepted until end of day today. I've only gotten 1 so far, and it was recall. No recall questions. Must be analytical. Must be using the information I'm teaching you. I don't care if you know how many nucleotides are in a codon. That's 201. We're in 409. We're using the information. But I guarantee by the end of the semester you will all be expert multiple choice question writers. Yes?
I do have a question. The exam is like a normal length exam. You don't make it longer.
No, it's a 50-minute exam. I'm giving them during our recitation period, so we don't have to worry about crowding, and we don't have to worry about time, because sometimes things take longer for some people. What else? Okay, so that's that. So, where were we? Yes?
How many questions?
1 million. I have no idea. My questions tend to be, I tend to go between 15 and 25 multiple choice and anywhere from 2 to 3 short answer, and there'll be less, but the value is equal on multiple choice and short answer. So you'll have 50 points multiple choice, 50 points short answer. Yes?
Is that in the application like lab scenarios?
It'll be use the information kind of question. Don't just spit it back. So I might ask you a question about how there's a gene that's being repressed, what kind of proteins might be involved, I don't know, something where you have to actually use the information. Okay. Any other questions about the upcoming anxiety-provoking exam? I know it's normal to be nervous the first time, but you can ask all the people in this class who had me for 408, and they'll tell you, oh, it's fine, right? Anyone? Someone? No? It's horrible. Okay. So, where were we? Let's learn something. So this is our little review slide. So people are starting to talk to me about study guides. Here's your study guide. These are the main points. Right? Big picture, chromatin structure and function. How is the chromatin structure? How does it relate to function? If you need to express a gene, do you need to do something to open up chromatin, to expose transcription factor binding sites, to expose the promoter? If you're trying to repress gene expression, are you more likely to do other things to your chromatin? So big picture. How post-translational modifications regulate protein-protein or protein-DNA interactions to affect the cellular function. So we did a little bit of harping on post-translational modifications because I love them, and now you guys love them too. So definitely understand acetylation, methylation, phosphorylation, and what their function is. Remember, we're changing the shape of a protein so that it can interact with another protein to do something. It's not just this magical thing that happens for no reason. It's all about regulation of gene expression. Then there was the concept of domains. This is a hard one. I've already been getting some questions. Remember, a domain is not a protein. It's a functional unit of a protein. Remember my Lego experiment where I took apart the Lego, like I had a bunch of Legos and I took different parts off. Those were the domains, right? If you remove a domain, you should lose the function that's associated. Yes?
So the chromodomain and bromodomain are attached to the protein.
They are a part of a protein. Yes.
They're building blocks.
You can think of it as a building block of a protein, kind of. Right, and it has a function. So a chromodomain binds to a methylated lysine on another protein. It's not a chromodomain itself comes in and does it. It's that part of the protein that does it. Okay? This is always really confusing for people. And please ask as many questions, come to office hours about this, because it's an important concept to understand, especially if you plan to do any sort of research in cell and molecular biology, because people are constantly looking at conserved domains. Okay, so we talked about chromodomains, bromodomains, and SH2 domains. So what do bromodomains bind to? Does anyone remember? Acetylated lysines. And so what effect might that have on gene expression? Activating, right? Usually acetylation is involved with activation, right? SH2 domains, we didn't spend too much time, but I did mention what do they tend to bind to? Phosphotyrosine. So that also it's about changing the shape of a protein to allow interaction with some other protein. If you don't get that phosphorylation or that methylation or that acetylation, there's something that's not going to happen. Yes?
Are fluorescents always used for tagging?
Huh?
Are fluorescents always used for tagging?
Is fluorescence always used for tagging? Is that what you're asking?
Is it like going back like the methods of
I'm trying. So can you give me an example of where the question came from?
we're looking at the northern blot.
The northern blots. So usually you will label a protein in some way so you can see it. Depending on your experiment, it'll be different. With the northern, you're having radioactivity. With immunofluorescence, which we're going to go over that practice problem again, then it's fluorescence. So you can think of it more broadly in when you're studying the function of a protein, you usually need to see it or its effect on something. You need to visually see it. And fluorescence is one method that you'll see. It's for tagging for seeing, yeah. It's not the biological process, but sometimes it might be a readout explaining the biological process. Okay. Then there are the enzymes that explain structure and function. Enzymes that regulate chromatin structure and function. So when you think about chromodomains, you should think about methylation. You should think about what kind of enzyme. What kind of enzyme is involved in methylation? Nope, that's phosphorylation. Kinase is phosphorylation. Methylation, methyl transferase. It's in the name. I know you haven't studied yet, but you know. Methylases, and then hats. What hats do? Acetylate. Okay, you guys got it. And then, of course, these are reversible, so there's also enzymes that remove these modifications. And then there are the ATP-dependent nucleosome remodeling enzymes, which we talked about on Monday. And those are also enzymes. And what do? They remodel nucleosomes, right? They either slide them around on the DNA or actually remove them from the DNA and reassemble. Or sometimes they'll remove one subunit of a histone and replace it with a histone variant. Then we talked about the concept of the histone code. Yes?
So with the ATP-dependent nucleosome remodeling enzymes, do they remove all of the histones?
Do they remove, you mean like all the histones in the whole genome at the same time?
No, like in the
Or-
nucleosome, all of the...
Oh, you mean all the subunits of the histone. It depends on the model that you follow, because I don't know if it's completely clear if the complete nucleosome is removed or not. But I do know you can swap out different subunits of the histones and replace them with histone variants. But there are models about whether or not you're just sliding around or actually removing the whole thing. So nothing's ever definitive in science. Concept of the histone code, right? That many histone modifications occur in those histone tails, and they mean something. And we're doing a paper on that in recitation this week. Those of you who read it already on Monday kind of got to see what that has to do with. And then histone variants. So the histone variants, remember these look like the same histone subunits, but they have different functions. So the example that we did on Monday, which we're going to start with right now, has to do with H2AX, which is a histone variant that is placed in histones in areas of DNA damage. And that's how the cell knows to recruit DNA damage repair proteins to sites of damage, right? Have you ever wondered about that? There are many ways that DNA damage is recognized, but one of the ways is through use of these histone variants. Okay, so let's go to this practice problem that we had. I know it confused a lot of people. So I want to orient everyone to what we're looking at, okay? We've got DAPI. That stains DNA, so that's your nucleus, okay? We've got CHEK2, which is a DNA damage repair associated protein. Don't worry too much about that. We have gamma H2AX, so that's the histone variant, and then the merge of all three. So anytime you see light, that means that the antibody or the stain is binding to the protein of interest. So here, all the DNA is being stained. So this is a nucleus. Do you think that nucleus is in mitosis? No. Good. Okay. That was a question just to make sure you guys were awake. Okay. So each little dot here represents a spot where the antibody against CHEK2 is binding to the CHEK2 protein. Okay. And same for gamma H2AX. So the problem was you're working at a pharmaceutical company and you're trying to identify new drugs for cancer treatment. These are the things you know because you have your biology degree from Towson. You know these things, right? A good cancer drug will cause DNA damage, which means cell cycle arrest and apoptosis. A lot of people got confused on this, so that's why I'm going over it again. People thought that we were trying to repair DNA damage and prevent apoptosis, but you need to switch your thinking when you're thinking about cancer. You're trying to kill cancer cells, right? We don't want them to survive, so if we repair, then they might survive. But anyways, so these drugs cause a massive amount of DNA damage, which results in cell cycle arrest and apoptosis in cancer cells. So then you cure cancer, right? So H2AX is the histone variant that is associated with DNA damage repair. So wherever there's DNA damage, you'll see H2AX. And same with, and CHEK2 is associated with that process. So this is a marker. These two proteins are a marker for DNA damage. If there's no DNA damage, there should be no staining here. This would be black and this would be black completely. Okay? So your job is to screen different compounds and then do high throughput immunofluorescence, which is not high throughput at all, and see if compound X is a good cancer drug. Okay? So we had a brief discussion in the last minute of class on Monday. So what did people think? Was it a good cancer drug or not? Yes. Why is it a good cancer drug?
DNA damage repair things were recruited to the DNA.
DNA damage repair.
So damage was caused.
Okay, so DNA damage repair proteins are being recruited to DNA. Damage is caused. Right. Did you want to add to that? No. Does anyone want to add to that, or is it?
Yeah,
is
that's
it well?
what I was confused. The DAPI cell is the one that has been treated. It's not the standard.
Yeah. So there's no control here, because unfortunately, I didn't have that figure. But a perfect one would have the same thing. This is one cell, and you took 4 different pictures of it. Well, 3, and then you merged them in 4. So in a perfect world, you would have another one where you have DAPI and then nothing here, which is not treated. Yeah, not treated. Any questions about this? Does everyone understand H2AX and the role of H2AX in DNA damage repair, histone variant? Anything about that? Yes.
So it can serve to actually repair DNA damage, but also it'll signal for apoptosis.
So, no. A lack of ability to repair DNA damage will result in apoptosis. So this is a marker for DNA damage. Yes, it recruits proteins to the site of DNA damage, but when you have overwhelming DNA damage caused by a really good cancer drug, then chances are you'll go the route of apoptosis. Very good question. Any questions about this practice problem? Yes.
Question, what exactly is the DNA damage?
What is the DNA damage? Well, we don't know because this is just compound X. Every protein might have a different function. Some proteins like to slide themselves in between the base pairs, so they interpolate. Some will actually bind to different bases of DNA, create these big bulky adducts. All these things are going to affect DNA replication, result in replication forestalling. Yes?
If we're dealing with cancer cells, wouldn't the ability to switch on apoptosis already be disabled?
You are smart. Yes. So the comment was, if you're dealing with cancer cells, won't the ability to switch on apoptosis be disabled? A very good cancer cell will probably have shut off apoptosis. But not like, so don't forget, apoptosis is regulated by multiple mechanisms, and some will be shut off. And I mean, yes, you're absolutely right, but not all cancers shut off apoptosis. Some do, and chances are you'll also arrest the cells, not only cause apoptosis, but you'll cause cell cycle arrest. So this, at least the tumor won't grow anymore. So you'll have, and then don't forget, when you're dealing with cancer, every patient is different. Every patient has their own different gene mutations or gene expression issues. And some people will respond to this kind of drug and some people won't. And you'll have resistance in some people. Yes?
Is there a way to turn on the apoptosis back?
Is there a way to turn apoptosis back on? Probably. There's probably some drugs that are trying to do that. And if you develop one, then I want you to remember me and give me lots of money because we talked about it here. That's the $1 million question, right? Can you fix the things that are broken in cancer cells, right? Yes?
Even if the cancer only apoptosis, won't that amount of DNA damage that is seen not allow it to replicate?
Right, so that's the thing. Right, two things. So even if a cancer cell turned off apoptosis mechanisms, then won't this amount of DNA damage not allow it to replicate? Exactly. So two things happen with massive DNA damage: cell cycle arrest, so no replication, and or apoptosis. So you could just have the cells, cancer cells are these rapidly dividing cells. If they're not rapidly dividing anymore, or not dividing at all anymore, that's still good in terms of cancer treatment, right? Okay, so before I move on, any more questions? I'm getting great questions today. Everyone's awake. I think 11 o'clock is the prime time for this class. I taught it at 9 and everyone was always snoring. I'm going to try in the afternoon next year. Uh-oh, I just got a note.
After
11
games,
o'clock.
you want to eat lunch.
After lunch.
Okay. Okay. We'll have to think about this. Okay, let's move on. So, what is this about? The histone code and chromatin condensation. Okay, so let's go back to the histone code. Does anyone remember what the histone code is? Yes.
The amount of modifications occurring within histone tails at the same, all at the same time.
Right, so lots of modifications occurring on histone tails all at the same time, and they tend to cluster and mean certain things. You'll have different kinds of modifications in situations of activation and different kinds of modifications in situations of repression. And so, and you'll all read more about it this week in our recitation. Right. So with so this is an example of the role of the histone code in chromatin, the histone code and nucleosome remodeling complexes in chromatin condensation. Right? Chromatin condensation means that tight packing of the chromatin, usually to silence things. Right? So remember we talked about all these different enzymes that are involved in modifying histone tails. Well, so in this textbook, in Alberts, he refers to those as writers, right? They modify the writing on the histone tail. And readers, which are these chromodomain or bromodomain containing proteins, come along and read that modification and then recruit other proteins to do something. So you can think about chromatin condensation as the spreading of this tight packing of chromatin, and it occurs through the collaboration of these readers and writers. So you'll get some methylation, which will recruit some chromodomain containing protein, which will recruit some nucleosome remodeling complex, which will then slide the nucleosome closer together, and it works kind of like that, right? So here they're showing there's this protein that binds to some region of the genome that recruits a probably methylase, right? Methyltransferase, and then that will result in recruitment of some reader protein, which will probably have a chromodomain, so it's binding to the methyl group, which will then recruit some writer, which will probably be some other methyltransferase, which will then modify the next histone, and then you'll get recruitment of this chromatin remodeling complex, which will help to slide this nucleosome over. And you start to get this over and over again until you get this spreading chromatin condensation. Okay, so that's how this can all work together. I put in a question that I answered, but let's see who is paying attention. What modification here do you think is being read? Thank you, methylation, right. Probably not acetylation. Why?
Acetylation is pushing them farther apart, not bringing them closer together.
Acetylation is more likely to push things further apart. Why?
So it can open up the
Because it tends to open up the DNA, right? Okay. Okay, so regulation. Don't forget, this doesn't happen randomly, right? How do nucleosome remodeling complexes know where to go? Yeah.
Is it by like acetylation?
Right. So exactly. You hit the nail on the head. This acetylation and methylation on the histone tails is recognized by some proteins that then come and recruit. So that's like the whole reader. The reader then will recruit in a nucleosome remodeling complex. And so there's one other mechanism. Yes.
I have a question. As far as acetylation and methylation, they only occur on the tails of histones?
No. So acetylation and methylation can occur on histone tails. So the question was about where acetylation and methylation occurs. They could occur on the core histone and on the histone tail. The histone tails, though, modification on the histone tails is associated with recruitment of these kinds of proteins that further modify the chromatin structure. But then you can have, so it's not only about more methylation and more acetylation when you move the nucleosomes around. There are a lot of other things that can be recruited to the chromatin, and and then also lysine acetylation on the core nucleosome is associated with that loosening of the DNA. So, yeah, it's not as simple as I make it sound. I was just talking to my PhD advisor yesterday. We're submitting a grant together, and I was like, oh, well, we'll just do this and this and this and this. And he's like, you can't submit that. People think you're so naive. You always think science is so simple. And I think that's probably what makes me teach well, because I simplify things. But it's really bad, I think, when it comes to doing real science sometimes, because I always think, whoa, well, it's this. It's just some acetylation, and you get gene expression. Well, it's not that simple. So always remember, it's much more complicated than I make it out to be, at least. So, anyways, right. So one mechanism of recruitment of chromatin nucleosome remodeling enzymes is through recognition of a modification on the histone tail. So that would be there would be some chromo or bromo domain containing protein that's recruited that would then recruit the nucleosome remodeling enzyme. The other option is you might have a protein that will bind to the DNA, which will recruit. So this is methylation, acetylation independent mechanism of recruitment of nucleosome remodeling complexes. Okay. Any questions about this? Yes.
So is that protein bind to a modification?
Modification independent, it's probably DNA sequence dependent. Yes, yes. So it could be modification dependent or independent. Yes.
So there's kind of editorial systems where they will incorrect protein binds to a tail that something we should want to not at all. So the incorrect one.
So is there an editing system to prevent the incorrect protein from being recruited to a histone, to a modified histone tail? Let's go back to the concept of the histone code, right? It's not just one acetylation. There's like 20 modifications happening on this histone tail, which creates a very specific pattern. So it's not just about one protein being recruited through a chromodomain or a bromodomain. It's actually a protein complex that's being recruited through multiple interactions with multiple modifications. So that increases the level of selectivity of what can be recruited. Now, can something be removed if it's wrong? I'm guessing. I mean, you have all these enzymes that remove modifications, but I don't know. Yeah. Yes.
About, you said how there's all these modifications on the histone tail. Well, if there's acetylation and methylation on the same one, will that like correspond with different locations, loosening and tightening, like at the same time?
Yep, so when you think about loosening and tightening, you want to think about the core modification on the core, because the acetylation of a lysine on the core histone is what interferes with that positive-negative interaction between the lysine and the DNA. When you have the modification with the tail, it's more about recruiting other proteins. There are probably many lysines on there are multiple lysines on histone tail, so multiple modifications do happen in different places. Any more questions? Okay. Okay, so we've got a practice problem. And you guys are going to take a minute to talk to your friends. You're sitting alone. Do you have any friends here? How about them? I had her last semester. You can take it, right? Yeah. So here's the deal, guys. Okay. You're studying a novel protein that's expressed by the XYZ virus. I learned my lesson in never putting a real virus name because people start to Google real viruses, like thinking that I'm asking about a real virus. So this is the XYZ virus, which Dr. Margulies can probably confirm is not a real virus, right? You're trying to characterize the protein's function and have the following information. You have the sequence of a protein and you blast it. Does everyone understand what blasting means? Right, so you blast it and you're comparing it to the database of all known proteins and looking for homology, looking for proteins that are similar. You find that it contains two conserved domains, a bromodomain and an ATPase domain. What is a likely function of your protein, and what experiments might you conduct to provide evidence for your hypothesis? So take a minute to talk, and we will reconvene.
So the
It's just as relevant as but it's probably so it's probably actually work. It's going to be suited to bromodomain is going to treat something that's it's going to do something. When you hyperlyze ATP, that's how you get to it. So it's going to cleave one pocket. Use the energy. So it's actually going to be doing yeah, it's going to do some remodeling. Probably. It's either because the remodeling complex are the ones that actually do the.
yeah, I know, but they got the
Yes, it's the remodeling complex. So So if the protein is promodomain, it's going to be probably slightly complex. It's probably going to activate. Oh, what experiment? Okay.
Okay, guys. I hate to bring up a rousing discussion. Did everyone come up with a hypothesis? Was there anyone who had no idea how to approach this question? I love honesty because I can help you there. Okay, if you're too embarrassed to raise your hand, please come see my office hours and we'll walk through it together if you don't understand after we go through it right now. I really like the way I saw most students in the class talking about it. That was awesome. Only a few people just were sitting there, and I'm going to assume they're just shy. Okay, so what is a likely function of this protein? I want to see someone who I never see. Someone who hates to raise their hand. I'm going to bring candy so I can throw candy at people who raise their hand when they never do. Okay, someone who always raises their hand. Yeah. Probably activates transcription. Okay, what brought you to that conclusion? Okay, so bromodomain binds to acetylated lysines, and ATP, from what you've been told in this class, ATPase domains are associated with movement of histones. So that will result in gene expression or transcription. So you gave me the answer, but you went like beyond. You didn't go beyond, but you skipped it and went to the broad idea. And I like that you're going to the broad idea. But there's a protein that does that, moving of nucleosomes and binding to acetylated lysines. What protein is that? Or complex of proteins?
I was going to say that's what we were talking about, the remodeling.
Remodeling complex. Right. Because what does a bromodomain bind to? Acetylated lysine. And right, if you're sliding around your histones, then you're probably remodeling your nucleosomes, right? So, what experiments might you conduct to provide evidence for your hypothesis? Now, for this, you have to think back to my Lego example where we were doing things with the different domains. Yes?
Remove one of them to see if the function still occurs, just tell whether or not.
So I heard remove one of them. Remove what? Okay, remove the bromodomain and see if what still happens. Gene expression. Okay, go back a step because we want direct function of this protein. We say it's a nucleosome remodeling complex, and we remove our bromodomain. It should no longer do what? Bind to what? To acetylated lysine and nucleosome. Right. So you should lose that interaction if that's the case. And yes, it should have probably a negative effect on transcription. And what else? I don't know if there's a way that we do this, but we would look up a method. What else do nucleosome remodeling complexes do? They move nucleosomes. We might want to see if the nucleosome is bound to a different segment of DNA. Oh, we learned about how you analyze protein-DNA interactions, didn't we? What experiment is that? Chromatin IP, great, yeah.
I was gonna ask a question, if you do use it for remediation, is there a way after you like link the DNA to the histone, the DNA that's the linking DNA, is there any way to count that?
I'm confused.
Like, you know how we use like the chemical linking to the histone for the DNA to lock it so that you could digest the linker DNA? Is there any way to count the base pairs for the linker DNA to see if you actually are opening or closing?
Okay, so the question is, okay, we chemically cross-link our histones and our DNA for our chromatin IP. Is there any way to count?
The base pairs between the histones to see if it is
Yeah.
opening or closing?
So basically count the distance between histones. I'm sure there is a way. I'm not sure of it. What's another way you can do it, though? What's another way you can map your fragments of DNA? Yeah. Sequencing, yeah.
Also, you could sequence the DNA around your histones, and then when you sort of take out your histones, you chop off that DNA, and then whatever you're left with is the DNA sequence in between the histones.
Oh, sequence your DNA that's attached to histones, and remove your histones, and then sequence
What's not associated with DNA?
what's not associated. Oh dear. Yeah, I guess we have to figure out how we're gonna get that. Okay, I'm hearing lots of good ideas. Any other questions? So anyone want to add to this? Because these are great ideas, but I'm always hearing from the same 5 people who I think that's great that they're really engaged. But what about some? I need a dark horse to come out with a brilliant idea. Where's my dark horse? No dark horses. Okay. Any questions about what this was about? Did we understand the role of the bromodomain and the ATPase domain in this question of what the point of that was? Right? What does an ATPase domain do specifically? Hydrolyzes ATP? Okay. Okay, let's move forward. So try it yourself. What is the likely function of the protein below based on the domain organization? What domains are present and what do they do? 1 point extra credit for those who emailed to me before Friday's class. This will be posted. You don't need to scribble down the sequence. Quickly, I'm not posting it. I'm going to delete it. No, it's going to be posted and you guys can take a shot at it. There may or may not be domains that you're aware of. Let's say you see domains you don't know of. You can look them up. There's a magical thing called Google, and there's also something called PFAM that you can look at, but either way, Google will do it. Okay. Okay, so let's move on. So direct methylation of DNA. So we've talked about methylation of histone tails. Question? No. Okay. If you have a question, so direct methylation of DNA. This is actual methylation of the DNA itself. You methylate cytosine, that's the C, and you end up with 5-methylcytosine because that's where your methyl group is added. And so this is accomplished by an enzyme called DNA methyltransferase. Right? Because it methylates DNA and not histones. Right? Because histone methyltransferase methylates histones. People frequently get confused about DNA methylation versus histone methylation. So I want you to keep that in the forefront of your mind as you study this. These are different things and they might have similar functions, but they're different and they're done different ways. So DNA methyltransferase has a tough job to do, right? Because what it needs to do, so here's your DNA, and here's your DNA methyltransferase bound around it, and here's your active site, right? And see this red right here? That is your cytosine. That's going to be methylated right there. And so notice, normally this is how DNA looks, where you've got your bases on the inside, right? Do you ever have your bases flipped out? No. Right, DNA is super stable due to this base pairing and base stacking. So the flipping out of that base actually requires a lot of energy. So where do you think the methyl group that is added to the cytosine, where do you think it comes from? Just guess. There's no reason you should know this. So guess is allowed. It comes from the methyltransferase. So it's kind of like a kamikaze mission. It's just there's actually a methyl group in the active site that is transferred from the enzyme to the DNA. And so as you would guess, you can't keep using this enzyme again and again, right? Because it loses that methyl group. Yeah, strange, huh?
Is there an enzyme that'll put it back on there?
Probably. Or I don't know. Actually, thank you for pointing out there's a gap in my knowledge. Let's make that a Stump the Teacher extra credit. What happens to this methyl group? Right, so the methyl group is removed from the enzyme and added to the DNA, so now it's missing a methyl group. Does that enzyme just die? Or is there another enzyme that comes along and adds the methyl group back on? Okay, so you guys can look that up. You have until noon tomorrow to get that answer. Reference answer to me. No, I think this. No, I'm Googling and sending you the Wikipedia link. A fully referenced answer. Thank you. Yes?
So does the DNA methyltransferase need to have ATPase domain as well?
Does the DNA methyltransferase have an ATPase domain? That is a great question, and probably.
Do we need a lot of energy to put that out?
Right, reactions that require energy need ATP usually, and they need to hydrolyze it. So probably does have an ATPase domain. I'm not saying yes because I'm not 100% sure, but that's a very likely possibility. Yes? You missed it. You can't sit in the back. No. Yes. The question was, is there a way of recharging the DNA methyltransferase? Because if the methyl group is being added to the cytosine from the enzyme itself, is that just the ends of the enzyme, or is there some other enzyme that comes along and methylates the enzyme? I don't know. Yes?
So for histone methyltransferase, is there a similar mechanism, or does that?
That is different. But now I'm gonna have to look at the mechanism. You guys are too smart for me. Yes, it's different, but I'm not sure. Yes.
Is it just that single cytosine that's flipped out, or is that part of the changing nature in the sense where all of it is vulnerable to the environment?
Okay, is the single cytosine flipped out, or is that part of the changing nature in a way? I think it's just that base that's flipped out because it takes so much energy because all the interactions between the bases keep that DNA structure so stable that it would take even more energy probably to break apart other parts of the double helix. Any other questions? Yes?
Does it get flipped back in now? Or does it
Yes, it gets flipped back in.
Can something read that it's methylated then if it's on the inside? This is not the point to know
Well,
if it's methylated or something.
Is it on, where is it, okay, so let's go back. So the question is, can someone see that it's methylated if the methyl group's on the inside? Is it on the inside? I mean, it's actually sticking out on the side. Yeah. So you can imagine that though the base comes back in, remember there's we didn't really talk about like the major groove and the minor groove. You guys have learned about this before, the major groove and minor groove. And you can actually access the sides of your bases from outside the DNA. Yes? What does a methyl group do? Yes, so let's keep going for the next 5 minutes. Okay, well, before I do that, DNA methylation patterns are inherited through the activity of another enzyme called maintenance methyltransferase, right? So you have these CG dinucleotides that are recognized and methylated, and then during DNA replication, remember you've got your parental strands that separate into your daughter strands and semi-conservative replication. Remember all that? So what this enzyme does is that it actually is directed to methyl groups, so it recognizes the methylated cytosines in the parental strands, and then it methylates the cytosine on the opposite side. So that's how you inherit this methylation pattern. There we go. What is the biological role of methylation? Yes?
Is methylation done only in one location or is it done all over the DNA?
Oh, it's done all over the DNA. People actually study methylation of whole genomes because it's really important in, well, transcriptional regulation, which is the answer to the question that's up there. Yeah. So what's the biological role of DNA methylation other than what I just told you? Does anyone want to expand on, or maybe people aren't paying attention? Yes?
It's a prevent replication.
To prevent
Well, it is a barrier in replication.
the barrier to replication. Uh-oh, I don't know. Is it?
Yeah, I think it is.
Replication. Okay, you might be stumping me, or I don't know. Send me a referenced explanation of that. I'm getting a note from Dr. Margulies. I don't think it's a barrier to replication because our whole genome is methylated like crazy, and we're going through mitosis. S phase and mitosis fairly frequently, so we would not survive as a species, I believe. But I would like to see.
Yeah, I meant transcription, sorry.
Oh,
Yeah,
transcription.
I'm sorry.
Okay, thank you.
My bad.
Much better. Yes, yes. So it's a barrier to gene expression.
Yes,
Gene
that's
expression.
what I meant.
Thank you. Thank you. And also thank you for pointing out the fact that it's easy to confuse replication and transcription. It's a common confusion. I just saw hands. Yeah.
Well, like on the whole, isn't this what the entire concept of epigenetics is based on?
Isn't this the entire concept of epigenetics? Yes. Who's heard of epigenetics? Yes, and I just had a rousing discussion the other day about it. Great. So what is epigenetics? Someone who raised their hand. So many of you did. So. Yes.
It's like the biological controls, like what's controlled not by the base sequence, but more like how the DNA is being regulated.
So it's a way of regulating gene expression not by the gene sequence itself, but by what? Yes.
Anywhere outside of the genome. So it's like post-translational modification.
Okay, so it's how modifications of the genome affect gene expression, and it's affected by the environment. Okay, what else? Yes?
Gene complexes working together rather than just genes.
Gene complexes working together rather than just genes. Okay, and why do you say that?
Because I feel like we know what specific genes do. We don't look at how genes work as a group that can affect certain biological.
Okay, so genes work as a group. If things are silenced as a group, that affects biological function, and that goes to the fact that often whole regions of DNA are methylated, so full groups of genes are silenced. What else? So a lot of it, a lot of it has to do with methylation of DNA and that being a mechanism for regulation of gene expression. So it has nothing to do. If you think about like the most basic understanding of gene expression, it's about proteins binding to sequences of DNA and recruiting RNA polymerase and gene expression happens. This is adding the effect of methylation, possibly inhibiting that from happening. Yes?
So this DNA methylation is directly inhibited transcription factors?
So now the question is, what does methylation do? Going back to the question in the back. Does it sterically inhibit transcription factors? That is one thing it does. Yeah. What else do you guys think methylation does? Recruits some new proteins. You guys see there's a theme in this unit. It's all about modifications, changing shapes of things, either recruiting or preventing recruitment of things. Right. Okay. I think we will, I see we've got 1 minute left. Let's stop here and we will start with the jump writing.
Final biotech.
question, why is that?
Yeah, why is that?
It's because the hydrogen bonds are all the other bases.
If, like, the bond,
It's in the book.
The guy where this is.
TC, we need to look at the structure.
So probably it's not. So.
Is there an I know, we can look that up. Yeah, and I appreciate Rob.
There was a question on quiz about DNA methylation, and then it's like a lot of the answers mention promotamines, and then how could you not do how could you forget my name 10 minutes after saying it?
I just want to tell you about a couple neat papers about methylation.